Welcome to the biochemistry laboratory for the CABNRfit camp. The first thing I need to show you is how to use a micropipette. And these are one of the most important tools you're going to use over and over again in biochemistry and molecular biology laboratories. They enable us to pipette small volumes, generally volumes less than 1 milliliter, down to 1 microliter. In today's lab, you're going to use two types of pipettes. You're going to use this one here, which is, we call this a P1000, and on the side it's written 100 to 100 microliters. That is 0.1 mil to 1 milliliter. And if you look on the front here, let's get this in focus there. There we go. So on the front here you can see it says 1000. So this is actually 1000 microliters. And you can adjust that down. So this, for example, will be 955 microliters. This here would be, let's see, 925 microliters. So this would be 0.925 milliliters or 925 microliters. And today we're going to be using volumes of 1000 and 2000 microliters, that is 1 mil and 2 mils. So you're going to have them set to 1000, that is 1000 microliters. The other type of pipette you're going to use is the smaller one. We'd call this a P200. And this one, written on the side, it says 20 to 200 microliters. And again, if you look at the dial on the front, this one right now is set to 200 microliters. That's the maximum volume you can do with these. And then you can wind this down. So today you're going to be using volumes of 200 microliters and 20 microliters, 200 and 100 microliters. So for your 200 microliters, set to 200. And to do 100 microliters, you're just going to wind this all the way down until it reads 100 on the dial. So there you go, there's 100 microliters. And you should always check with pipettes you're using the correct one. So this one is a 20 to 200 microliters, and that is now reading 100 microliters. We dispense liquid with these micropipetters using micropipette tips, because we don't want to just stick this into our liquid, because that would mean we'd be cross-contaminating the whole time. So you'd be mixing up your chemicals, you'd be contaminating things with bacteria, you'd be doing whatever. So we use these micropipette tips, and we use a clean one for each new solution that we're going to pipette. So we make sure that our pipette is set to the correct volume. You push the pipette onto a pipette tip, like that. Just push it down and you'll see it should be firmly seated on the end of the micropipette. Close your pipette tip box, get it out your way. Now we're going to put liquid into the tip. So again, check you got the right volume. Wrap your fingers around the pipette like that and use your thumb to depress the plunger. So you push the plunger down to the first stop that you can feel. So just down all the way. Then you put your pipette, and you see I grab hold of the thing that I'm going to pipette in and out of. So you put your pipette tip into the liquid. Push down the plunger, pipette tip just below the surface of the liquid, and keep the pipette vertical. That's really important. And then slowly let the plunger come back up, and that will fill your tip with liquid. You then look at it. You say, okay, does that look like 1 milliliter to me? And make sure that there are no air bubbles in the volume that you just put into the tip. Then you pick up the thing you're going to pipette into. And this is really important. You don't want to just have things on the bench and be stabbing at them with the pipette, because you can knock things over really easily. So you always want to have one hand on the thing you're pipetting into. So then, put the tip of the pipette against the wall of the thing you're pipetting into and push the plunger, now this time all the way down, and really push it all the way to the end, and that'll blow out any remaining liquid. Okay, let's just go through that quickly once more. Check you've got the correct volume on your pipette. Push the plunger down to the first stop. Put the pipette tip with the pipette vertical into the liquid, just below the surface of the liquid. Allow the plunger to come up slowly. Take the tip out of the liquid. Check that you've got the thing properly filled. Then get the thing you're going to pipette into, put the tip against the wall, and then push the plunger all the way down to the second stop to dispense any liquid. Now that is how you use a micropipette. So now you know how to pipette, you're ready to do your experiment. And for our experiment, we're going to be mixing together our cholesterol assay mixture. So this assay mixture contains all of the enzymes we need, that if we add cholesterol to this mixture, we'll get the purple colour, which we're then going to measure in the spectrophotometer. So there's our assay mix. We have a series of solutions in the ice bucket here. We have our cholesterol standards, so we have one at 10 mg%, we have one at 20 mg%, another one at 30 mg%, and finally our unknown, where we don't know what the cholesterol concentration is. You'll actually be given smaller volumes. You'll be given little Eppendorfs to work with. The other thing we have is some isopropanol here in a bottle, labelled isopropanol, and that is for making your standards, or for making the zero rather. Okay. You will also have, and you'll have this printed off, is the pipetting table for setting up this assay. So this will be in the materials on Web Campus, and we have a number of columns here. So what is the sample, how much of the sample do we add, how much cholesterol assay mix, and then there's a column here for recording the absorbances of the samples. We use this as our guide for what we're going to pipette. I'm going to pipette everything into these disposable glass test tubes here. So I've already got these set up and I've labelled them. So I have one tube for the 0, and that is labelled 0. I have duplicate tubes for my 10 mg% sample, labelled 10, duplicates for the 20, duplicates for the 30, and duplicates for the unknown. So in total I have 2, 4, 6, 8, 9 tubes. Unknown, 10 mg in duplicate, 20 mg in duplicate, 30 mg in duplicate, and the unknown in duplicate. So they're all labelled and ready to go. So I'm going to start by pipetting in my samples. For the 0, I'm going to pipette in 200 microliters of isopropanol. And this is isopropanol because our cholesterol standards and unknowns are dissolved in isopropanol. Cholesterol is a very fatty, a very hydrophobic substance. It doesn't dissolve well in water. So we're going to start with 200 microliters of isopropanol. So we set this pipette, we set our pipette, our 20 to 200 microliter pipette, to 200 microliters. There we go. Remember, so this is the 20 to 200. And this one uses a yellow tip. So we push a yellow tip onto this pipette. We then get our isopropanol. And we pipette 200 microliters of isopropanol into the tip. I'll just show you that one more time. So push down the plunger, put the yellow tip into the isopropanol, let it come back up, close off the bottle, pick up the correct tube, so this is the 0 tube, and pipette 200 microliters of isopropanol into that tube. You then dispose of the tip, so we have waste baskets, so you can just dispose of the tip. You simply push that plunger and the tip will pop off the end, but make sure you do that into one of the dry waste baskets. We're now going to pipette our 10 milligram percent, 100 microliters of that. So we're going to need to reset the pipette to 100 microliters. There we go, so that's now set to 100. Get our 10 milligram percent sample, and we're going to pipette 100 microliters into each of these two tubes, like that. And once I add, I'm actually going to move the tube so I can see where I am along the row here. I know that I've made an addition to those tubes, so I'm just going to move them down the row. I then get a clean tip. You change the sample, you change the tip, and now pipette my 20 milligram percent samples. Again, 2 of these, 100 microliters and 100 microliters. Change the tip, new tip, goes along, and now I go to my 30 milligram percent. I'm going to go into these 2 tubes, so into my 2 30 milligram percent tubes. Finally, the unknown, 100 microliters of unknown in duplicate, so into the 2 tubes labelled U for unknown. So 100 microliters, 100 microliters. Now we've got all of our standards and unknowns, we're ready to add our cholesterol reagent, and we are going to add 2,000 microliters to this tube and then 1,000 microliters to all of the other tubes. We need double of this, you'll see when we use a spectrophotometer, because it goes in the front and the back. So we go back to our 1,000 microliter pipette, have it set to 1,000, and we put a clean blue tip onto the pipette. So we're going to pipette 2 times 1,000 microliters into our 0 tube, like that. Pipette and mix them all together. Then in all of the other tubes, each tube will get 1,000 microliters of our assay mix. So 1,000 there, move the tube. 1,000 there, move the tube. And you notice I keep this pipette vertical. You do not pipette with the pipette at an angle. You keep it as close to the vertical as you can. Mix. Actually, I'm just going to mix all of these once they're all pipetted. So 1,000 microliters into every tube. Okay, so every tube now has some of the assay mixture in it. I'm going to mix them, just kind of flip the bottom of the tube like that. And you can actually already see that the tubes are beginning to change colour. So the 3 enzymes in this mixture are converting the cholesterol into products, and hydrogen peroxide is generated, which the final enzyme takes the hydrogen peroxide plus some substrates and makes this pink colour. Now you may be able to see, so for the unknown it's still this kind of brownish colour, and for our standard it's already developing some pink colour. So what we do now is we take these and we're going to incubate them at 37 degrees Celsius, so that's going to speed up the reaction. These are mostly human enzymes, and they work best at our temperature, which is 37 degrees Celsius. So once you've pipetted everything and mixed it, then it's going to go into the water bath for 15 minutes. After 15 minutes in the water bath, your samples will have developed some colour. So the amount of colour that you get, the more cholesterol that you add to the assay, the stronger the colour you're going to get. So you can kind of see this, that if you look at the 0, it's pretty pale. You look at the 30 milligram percent, it's really quite a strong pink colour. Now, because the amount of pink colour is proportional to how much cholesterol we added, if we can figure out how much colour we have, that is by measuring the absorbance of our samples, then we can relate that to how much cholesterol was added in the first place. And to do that, we're going to use this instrument here called a spectrophotometer. And the spectrophotometer shines light of specific wavelengths. You choose the wavelength to shine through depending upon the nature of your sample. And then it measures how much of that light gets absorbed. And it then reads out here on the computer as an absorbance value. The first thing you need to do is to zero the spectrophotometer. Now, don't worry too much about all the details here, because when you get to this in the lab, we're going to be working very, very closely with you to help you to use these machines. But the basic principle here is that the more color you have, the more absorbance you will get, and we're going to measure the absorbance using the spectrophotometer. The first thing we need to do is to zero the instrument. We need to tell it what no absorbance looks like. And to do that, we use the sample we set up where it's just the cholesterol assay mix and isopropanol, that is our 0. And we're going to pour this into 2 of these little plastic cuvettes here. And again, we're going to work closely with you to make sure that you use these correctly when you get to it. But if you look at them closely, you'll see that they have a clear face and an opaque face. And the clear face is the one that's going to be facing the beam, which is actually running parallel to you if you're standing in front of the spectrophotometer. So, we take our 0 tube contents and we pour it equally into 2 plastic cuvettes. Just pour a little bit into there. We check that there are no air bubbles, and we can also wipe off our cuvette using a piece of Kim wipe, just to make sure there are no air bubbles, make sure there's no smudges on our cuvettes. Our samples go into the spectrophotometer here, and again we're going to show you exactly how to do this. We put both cuvettes in the correct orientation. Close the lid, and on the computer screen here we hit auto 0. What that then does is it's telling the machine what 0 absorbance should look like, because there's no cholesterol in this sample, we shouldn't be getting any absorbance. Once we've zeroed the instrument, we take out the front cuvette, pour its contents back into the 0 tube. We can then rinse out the cuvette. We do have some water bottles, so you can just rinse the cuvette very briefly. And now you're going to start adding your standards and unknowns and reading their absorbance. So to do that, we start with our 10. Just pour it into the empty cuvette. Put it into the front of the spectrophotometer, close the lid, and there's a window here, and this is reading the absorbance of this sample is 0.239. And I'm going to write that down on the table that I use for the pipetting, so 0.239. We'll then take this sample out, and you can pour it back into the tube. Then take our second 10 mg% sample, pour it into the tube, it goes into the spectrophotometer, read the absorbance here, and it should be very close to the absorbance of the other one because these are duplicate samples, and this one reads 0.219. So maybe my pipetting wasn't absolutely perfect. The closer that you get the closer those absorbance values are to one another, then that's telling you that your pipetting was very good and very reproducible. We take that sample out, pour it back into its tube, and then we continue reading. I'm going to read the 220s and the 230s. Now, I don't actually need to rinse out the cuvette. When I'm doing the standard, as long as I start at the least concentrated and go to the most concentrated, I can just kind of get any drops out of the tube and just pour in the next one. So here's our 20 mg% sample, and this should have a higher absorbance than the previous one, and it does indeed. Its absorbance is 0.401. So I'm going to write that down. I'm going to keep doing this. So pour that out. Pick up our second, our 20 mg% duplicate. Close the lid. Look at the absorbance. Okay, that's 0.391. Write it down, and so on. So I'm going to continue doing this until I have the absorbance measurements of each one of my samples. Like I say, I record all of these in my table here, so I write down the absorbances of each one, and the last thing I'm going to show you how to do is how to turn this into a standard curve and to figure out the absorbance of our unknown.